Hi, my name is Paloma Bravo, and my research project was over cell communication in fly spermatogenesis. It was conducted in the T.H. Morgan Department of Biology at the University of Kentucky, and my mentor was Dr. Douglas Harrison. So the JAK-STAT pathway mediates many cellular responses to cytokines and growth factors, and it's found in a wide variety of mammals like humans, and it's also found in fruit flies. Now, in fruit flies, this pathway is a lot simpler, but it's still a great model to test the JAK-STAT pathway in. And individualization is a really prominent step in differentiation of spermatids or the maturation of the spermatids. So during this individualization, the 64 interconnected spermatids that are in a single cyst push unnecessary cytoplasm out and individualize. And after this, they travel to the seminal vesicle where that's their final destination. And it was actually found that the JAK-STAT pathway is required to activate the initiation of individualization. And the way the JAK-STAT pathway does this is by regulating both somatic and germline cells. And those two work together to initiate individualization. And here I have a diagram of spermatogenesis. And on the left, it shows the testis and the different structures. And the maturation of spermatids actually begins in the testis hub, and it travels up as the spermatids elongate and individualize. And in order to individualize, the investment cones or individualization cones, which are shown in red on the figure on the right, actually start at the head of the cyst cell and then push all the unnecessary cytoplasm to the back and then dispose of it in a waste bag. After this is done, the 64 spermatids individualize in the trapezoidal seminal vesicle where they can be used. Now, the goal of this research project was to identify candidate genes in both the germline and the cyst cells that, when are regulated by the JAK-STAT pathway, can initiate individualization. My hypothesis was that the JAK-STAT pathway is affecting individualization by directly regulating signal production by transcriptional activation in candidate genes and by either regulating somatic cyst cells or germline cells or both. In order to test this hypothesis, the flies carrying specific genes or like the knockdown of genes were bought from a laboratory that specialized in making these flies. And the testes from these flies were dissected and stained in a phalloidin solution for about an hour. And this phalloidin solution contained DAPI stain and phalloidin stain in a ratio of about 1 to 500 microliters. And in order to analyze the result, the phalloidin stain stained individualization complexes, or ICs, which were used to see the number of spermatid bundles that were going or had gone through individualization. And the DAPI stains DNA, so it was used to score how many total spermatid bundles were under each frame. And both of these were scored under an LED microscope under different light settings. Right here is a picture that I took when I was scoring the total number of nuclei under the DAPI light or the violet light of the microscope. And since DAPI dyes DNA, it's very hard to pick out the elongated spermatid bundles because there's so many structures that contain DNA within the testis. So the easiest way to identify an elongated spermatid bundle is by looking for an oval shape that is filled with lines within, which are the spermatid bundles. So right here where I have an arrow pointing are around 3 elongated spermatid bundles. And it's a little bit hard to see in the picture, but when you look a little bit closer, you can see the individual lines of spermatids within the spermatid bundle. And the same was done for the phalloidin under the phalloidin light setting. And these spermatid bundles actually look the same. They're just under a different light setting. And when counting for the individualization complexes, both basal and non-basal individualization complexes are counted. The difference between these two is that basal can be seen under both phalloidin and DAPI light, while non-basal can only be seen under phalloidin light. The difference between these two is that non-basal shows that individualization has been going on for some time and is nearing the end, while basal individualization complexes have recently started. And although both of these text individualization, they're just counted separately because of that. And for the results, so each gene was crossed with either Vasa or Aya-gal, and Vasa drives expression in somatic cyst cells. So if a particular gene, when crossed with Vasa, were to individualize, that specific gene is regulating the individualization through somatic cyst cells. And when these genes were crossed, they were compared to the gene W, or the cross between Vasa W, which is the first column shown on this graph. And gene MMP1 and HP-TIMP have no individualization complexes, neither basal or non-basal. So those genes do not regulate somatic cyst cells. And then for the gene MMP2, although it is a little bit less, there's a lot less individualization complexes as the control, there is still a pretty significant amount of individualization complexes, which means that it does have a role in regulating somatic cyst cells. And Aya actually drives this expression in the germline. And all of these two genes that I tested were crossed with Aya to test the regulation, to test the individualization, whether or not it would happen when compared to Aya crossed with W. And the numbers are actually pretty similar to the control group, which means that both MMP1 and MMP2 had a pretty prominent role in regulating the germline cells, which then initiated individualization. So to conclude, two genes that mediate the initiation of individualization were found, partially supporting the goal of this project, since the original goal was to test around 300 genes from the candidate gene list. And MMP1 and MMP2, both of them regulate the germline cells, but MMP2 also regulates somatic cyst cells, which gene MMP1 does not. And HP-TIMP was only tested for somatic cyst cells because I only crossed it with Vasa, and it was not shown to have any effect, which means it does not regulate somatic support cells or somatic cyst cells. And I did not test it for the germline, so that would be one of the next steps that I would like to take with this experiment. So there are a few sources of error in this experiment, the two biggest being the limited amount of trials and the lack of a statistical test. This was done only after one trial, so there would definitely need to be more trials to make this project a lot more reliable. And there's not a statistical test that I found that would allow me to give a statistical validity to the results. So that's also one of the next steps that I will need to take to make the results of this project more valid. And some more steps are to test more genes. So I only tested 2 or 2.5, I guess, of the 300 genes from the gene list. So I would like to test more genes to take this project further. And also testing for both soma regulation and germline regulation by crossing all of the genes with Aya and Vasa, since for I think HP-TIMP, I was only able to test for somatic regulation. I think it's really important to test both to see the full effect that each gene can have on the individualization. And finding the specific genes and the relationship between the genes in the JAK-STAT pathway and the soma-germline relationship in individualization is important like finding it in fruit flies, because it can be translated to humans, and understanding further developments of the JAK-STAT pathway. And it can help solve a lot of like human problems like infertility or like growth factor deficiencies. So a few people and programs were able to make this project happen. To begin with, my mentor, Dr. Douglas Harrison, my lab trainer, Sepida, the University of Kentucky, specifically the T.H. Morgan Department of Biology, the Kentucky Young Researchers Program, and Miss Young and Dunbar High School. Thank you.